Sounds
Okay, hello everyone. We're going to be going over the dystrophin protein. It's from me and my partner, Chris Caldwell. We're going to give a quick background to like why we chose a specific protein and everything behind that. So I was doing studies on this disease called Duchenne's muscular dystrophy, and I saw that it has to do with the lack of dystrophin protein. So dystrophin protein specifically has to do with our muscle cells, and Duchenne's muscular dystrophy leads to a lack of the entire dystrophin protein. There might be other diseases where there's partially functional dystrophin, but this one has no dystrophin at all. And this leads to poorly nourished muscle. So it's not going to function as well as like probably normal muscle cells or just even the muscle organ itself. So here we're going to go over the dystrophin protein and its complex with intracellular actin and the dystrophin-associated protein complex. So on the left, we have a basic image. We see dystrophin binding to intracellular actin and a muscle cell membrane, which we're going to call it the sarcolemma, since it's the muscle cell. So what the dystrophin does with its binding to intracellular actin and the dystrophin-associated protein complex, DAPC, or on the right, DAGC, it acts as a cellular scaffold. So what it does, it acts as a scaffold by absorbing the mechanical force generated from the intracellular actin and helps the muscle cell move with the actin as it's doing its contractions. So it helps us sort of like the shock absorber or force absorber. And its main function is to make sure the cell doesn't stretch too far apart and tear and cause holes, specifically from the mechanical force from the intracellular actin, and helps pull the membrane along with the force to act like a shock absorber and promote like muscle membrane fluidity. As you know, like a rigid muscle cell or just rigid cells in general are prone to just causing pores, breaking, and leading to muscle cell death or just cell death in general. So here is a more macro and large picture of the muscle cell and its location of dystrophin. As you can see on the left picture, the membrane is the location of the dystrophin, is part of the muscle cell, and is part of this entire muscle cell fiber. So you can see how important dystrophin is in the entire organ system. It helps with the force generation and also the force absorption and also being able to generate force through your entire body. So without dystrophin functioning, your muscle cells aren't going to be fluid with their ability to generate and sustain force. And if this leads to some muscle cell death, over time, you're gonna be able to generate less and less force, as you can see with the muscle fiber here with the sarcolemma. So here, we're gonna go over the top image first. It's gonna be pretty much going over the domains for dystrophin. So the left side has the actin binding domain, and then we have the rod domain, the dystroglycan binding domain, and the dystrobrevin or CT domain. So on the actin binding domain, it's considered the N terminus. So the N terminus is where you bind to the actin. It establishes that strong connection to help with scaffolding. The rod domain is pretty much the entire domain that stretches from the N terminus to what we call the dystroglycan binding domain, the WW or the CYS. So it's pretty much just a bridge to get over there, or like a metal beam. And that beam is going to attach to the WW, CYS, and those two regions specifically have to deal with binding to the sarcolemma. So the CYS is called the CYS region because it's rich with a bunch of cysteine, which helps its embedding into the sarcolemma. And the WW domain is interchangeable with the cysteine-rich domain when people are talking about it in studies. This dystrophin protein interacts with many other proteins around the CYS and WW region, and it's going to be called the dystrophin glycoprotein complex, or we'd call it the DCG. This entire complex has just a bunch of proteins that stick together to help maintain structure between both the sarcolemma and also the junctions all around the muscle cell. Over to you, Chris.
Yep, thank you, Anthony. So electron microscopy can be used to detect the presence of dystrophin in muscle tissues, as shown here in the picture, which is a dystrophin molecule in gizzard muscle cells. However, electron microscopy cannot be used to find the structure of dystrophin. This is likely due to many regions of dystrophin being embedded in the DAG complex and interacting with the DAG complex, which prevents the isolation and penetration of the specimen. However, X-ray crystallography can be used to produce images of certain regions of dystrophin, but not all of them. It can produce images of the N-terminal domain, the WW domain, and it can also be used to find the structure of the N-terminal first spectrum repeat of dystrophin. So the other regions cannot be determined through X-ray diffraction because of the same reason that you can't use electron microscopy to determine the structure of these regions, and that's because it is too embedded in the DAG complex. However, we can use utrophin, which is a related protein to dystrophin that is also in the DAG complex, because it shares a lot of homology with dystrophin. So scientists can study utrophin to assume the structure and function of dystrophin from what they find from utrophin. And utrophin's structure has been identified with the use of cryo-electron microscopy. When comparing dystrophin to utrophin, they both play a role in the DAG complex and share the same function at certain points. However, the way that they interact with the complex varies. So in the top image, this shows dystrophin and its interactions with the DAG complex, and the bottom image shows utrophin's interactions with the DAG complex. A little bit more about utrophin. It's present in fetal development, and its interactions with the DAG aid its function to stabilize the neuromuscular junction as you get older. Whereas with dystrophin, it's present after fetal development and is able to dimerize itself.
So yeah, pretty much as you get older, like dystrophin just replaces utrophin as it's a more stable and compact structure.
So the first structure in dystrophin that has been imaged through X-ray crystallography is the N-terminus region, which is responsible for binding to F-actin. As shown here on the left, this domain from dystrophin is comparable to the image on the right, which is the domain found in utrophin. So one of the only differences between the two here is that there are different residues between the two helices of the protein, which adds another kink to dystrophin's N-terminal domain. This gives dystrophin a more rigid structure compared to utrophin's more curved structure. So this similar structure of the two proteins allows them to both interact with F-actin in a similar function. However, this different function, the slightly different structure of the two proteins, allows them to perform slightly different functions that were previously mentioned on other slides. So the second structure identified with X-ray diffraction is the N-terminal first spectrum repeat. This is the 3 helical bundle structure that is present within repeats in both dystrophin and utrophin. So 24 of these spectrum repeats separate the carboxyl and amino terminals in dystrophin, with the first spectral repeat making up a part of the N-terminus domain. So there are many more spectrum repeats, however, they make up more of the rod domain, and they are believed to interact with other proteins of the DAG complex, such as Dystrophin. In this image, A displays the first spectrum repeat in utrophin, and C displays the first spectrum repeat in dystrophin. And the structure of these first spectrum repeats are the only structures found of the spectrum repeats. The other spectrum repeats within the rod domain have not yet been determined. So this is the WW domain, which is the only other domain of utrophin or dystrophin that has been mapped. So the green is the actual WW domain of dystrophin, while the blue is the specific peptide within the DAG complex, which is also known as beta-dystroglycan. This complex is what helps dystrophin attach to the sarcolemma, but we don't have too much information about this because we can't really access the internal structure as is embedded in the DAG complex, as mentioned before, and these hydrophobic interactions prevent us from acquiring more information about it. So the only info we do know is that the cysteine-rich domain bonds very tightly to the sarcolemma due to having many different disulfide bridges made, which help it live up to its role as a scaffold by having a very sound base.
We're gonna go into a little bit more information on the N-terminal domain. The N-terminal domain is it dimerizes with itself to make up the CH1 and CH2 domains. As you can see, they're both pretty similar and they're actually exactly the same. They're just 180 degrees flipped on each other. So then these two domains work to create the active site of dystrophin shown below. And these are the important residues for binding with actin, as we can help figure it out using the information from utrophin before. And with those specific interactions studied, we have determined that GLU65, ASP46, and ASN75, which are all around the central alpha helix and the C-terminal and N-terminal domains of the specific N-terminal protein, part of the protein, are important with its F-actin binding.
And from the information that we know from the N-terminus region active site for dystrophin, we don't know too much about it, but we can use the homologous active site of utrophin when it interacts with F-actin, which there is data available of it actively interacting with actin. So we can use that to assume the function of the active site in dystrophin. The top image shows the same image from the previous slide and mentioned before with the key residues for the dystrophin actin interaction. And the bottom image shows the interaction between some residues that are present in utrophin, which are colored yellow in stick formation. And these interact with the F-actin, which in this image is the magenta colored.
So yeah, we're gonna go over the importance of this entire dystrophin protein and its entire effect on pretty much the entire organ and organ system, and I guess entire organism as we go up along the chain. So here we're gonna look at the soleus muscle. The soleus muscle is a very easy muscle to study because it's involved in walking, transfer of force, absorption of force, especially for things like walking, running, and jumping. So there's a study that was made to study the effects of dystrophin in mice on their soleus muscle, as they have pretty much similar functions. So on that image on the right, we have A and B. A is going to be a regular soleus muscle of a mouse, and then B is going to be the soleus muscle after 4 weeks of development. So A is around 3 days to 1 week, and then B is around 1 week. Same for A prime and B prime, but A prime and B prime are muscles of mice that have dystrophin mutations, so they have no functional dystrophin. As you can see in A and B, you have a healthy soleus muscle, and then you can see a little darker staining on B, representing hypertrophy or the muscle cells getting bigger. As you can see, it's more developed and has a little deeper staining compared to when you go from A prime to B prime. You have a good bit of staining, but some dark spots. And then you see B after 4 weeks, like there's a bunch of these muscle cells that don't have nuclei anymore. So that means they die, as you can see with less staining. This is specifically important when we transfer that to humans. We have Gowers' sign and large calves. So individuals with DMD, Duchenne's muscular dystrophy, no functional dystrophin protein, have enlarged calves, and you'd wonder why. Why would they have enlarged calves instead of smaller calves? Because their muscle cells are dying. This is because these muscle cells are being replaced with fat cells. Those fat cells from the body are there just to help with base of support, increasing the mass of our legs so we can try to stand up a little easier. As you can see on the left picture too, we have this thing called Gower sign for children with DMD, where they have to use their hands to push on their legs to stand. This is due to the inability of their lower leg muscles to generate force to stand. So they need to find some other way to increase spatial support. So this happens around 5 years old. And then this is pretty much the prognosis of DMD on how it affects the entire body. At 2 years old, you're okay. But at 5 to 6 years old, you're gonna have like weaker leg muscles. You're gonna have like this arched back. That's gonna be called that's what we'd call lordosis. And around 15 years, your lower leg muscles can't help you stand anymore or do normal function. So you're gonna be in a wheelchair the entire time. And then after that, your body is going to have other issues with not just your lower leg muscles, but the rest of your muscles in your body, specifically the heart. So your heart's going to have trouble pumping blood. So people die around 20 to 30 years old from DMD due to just heart failure or other heart problems. So yeah, this is our presentation on dystrophin and how it affects the entire body, going from molecular all the way to macro. Thanks for listening.